Good morning, students. Last time we were discussing this structure of the tRNA, and we have last time discussed some of the characteristics of the transfer RNA, or we can simply call it as a tRNA. We know that this tRNA is a single-stranded RNA, but due to what we call as the too much foldings, it seems to be the double-stranded RNA. It is the smallest form of RNA and it is mainly made up of 75 to 90 nucleotides. There are mainly 20 types of the tRNA. It is mainly because of the 20 different types of the amino acids. अगर हम बात कर लेंगे molecular weight of this tRNA, it is between 25,000 to 40,000 Daltons, and we have also discussed that it is also called as the soluble RNA because it is soluble in the 1 molar NaCl solution. It is also called as adapter RNA because it carries amino acids to site protein synthesis. And what we'll see, जो हम इसका स्ट्रक्चर पढ़ लेते हैं इस ट्रांसफर आरएनए का, इट इज कॉल्ड एज अ क्लोवर लीफ मॉडल. एंड दिस मॉडल की अगर हम बात करेंगे, इट वाज गिवन बाय रॉबर्ट हॉली in the year 1965. Now what we will see, जो ये क्लोवर लीफ मॉडल है, इट कंसिस्ट्स ऑफ सम एबनॉर्मल और वी कैन से सम मॉडिफाइड बेसिस, abnormal or we can say some modified bases. And these bases जो यहां पर पाए जाते हैं, these are number first pseudo uracil is one. Second one we have got the inosine. Third, what we will call, we have got dihydrouridine. These are the some abnormal or 3 abnormal or modified bases which are found in this clover leaf model. And this what we'll see altogether, what we will see in this model, there are 5 arms, and out of which 5 arms only 4 arms are important. Those are important. Now what, जो इसका डायरेक्शन जो है इस क्लोवर लीफ मॉडल का, इट इज डायरेक्शन इज इन 5 प्राइमर to 3 prime. So let us first draw this structure of this, then we will try to discuss model. First let us try to draw the structure of this model. This is the structure and these are the 5 arms, 1 2 3 4 and 5. Now if agar we will write the direction of this, ये कंस्टेंट होता है 5 and 3 prime, this 3 prime. Now actually it is a single stranded, but when we see the diagram of this, it seems as if it is the double strand. It seems as if it is the double strand. but actually that is not the case. It seems as if it is the double stranded, and this double stranded is mainly due to too much foldings, that it is seems to be the what we called as this double stranded. And what we will see कि जो यहाँ पर हम क्या देखते हैं कि that बीच में जो यहाँ पर हम देखते हैं कि इन दीज आर्म दिस इज द लूप 1 2 3 एंड देयर आर 5 arms. This is the one arm, second arm, third arm, fourth arm, and fifth arm. Now here we will see these are the nitrogen bases, these are present, and these nitrogen bases के दरमियान क्या देखते हैं, जो bonding है, there is too much bonding between these nitrogen bases. हाँ, again what we will see, nitrogen bases. These are the nitrogen bases, these darker line. Now at what we see, there are altogether 5 arms, and out of these 5 arms only these 4, this one, this one, and this one, and this one, they are important. But this is not too much important, this one, because this has a small projection here which is considered to be what we called as this vestigial arm, and this is a variable arm. Rather, this one, it is a variable arm. It is considered to be vestigial, that is, it is of no importance to this tRNA. Now अगर हम ये देखते हैं कि what we will see at 5 prime end, what we will see, जो यहाँ के हमारे पास जो base what we have got there, this is the G यहाँ पर. हाँ, at what we will see, a special sequence is always present at this 3 prime end, and it is the CCA. This code, this is the codon, this is the CCC code, and then we will see there it is the functional group, and that functional group is OH group. Now this is the first arm. This first arm is called as, if we call this, it is called as activated, this is called as activated amino acid attachment site. Amino acid attachment site. See here what we will see, this again जो यहाँ पर है amino acid, it gets attached at this place. So if we draw this diagram, we will find this like this. In this box what we will see, जो हमारे पास जो amino acids है, it get attached at this site. I will write in the abbreviated form AA. This is the amino acid. It gets attached at the site. We can simply call it as the amino acid binding site. Now, this second arm, अगर हम बात कर लेंगे, you will see here there are almost 7 bases in this system. अगर हम बात कर लेंगे, हाँ, इसको हम बोलते हैं this is called as a system, this is called as a system, this is called as a system, this is called as system, this 2 is called as the system, this 2 is called as system, this 2 is called as system, and what is this? This is called as loop. This is called as loop. Now there are the bases present in these arms, and these bases जो हैं what we will see, it is that place where the hydrogen bonding takes place. और जो loop है, this loop and this loop, it is that place where we will see no hydrogen bonding takes place. Whereas in the arms, hydrogen bonding takes place. In the loops, we will see no hydrogen bonding takes place. Now this what we call as this arm, इसको हम क्या बोलते हैं? This arm, this is called a second arm, rather this is the second arm. अगर हम यहां पर लिखने की कोशिश कर लेंगे second arm, it is called as T psi C. This T, it is the thymine, but it is the only arm where we will see it is a thymine. It is actually the RNA. So instead of the thymine, we must have got this uracil. This, what we call as this psi, it is the pseudo uracil. This psi, it is the pseudo uracil. It is the pseudo uracil, or we can say it is this abnormal base pair. And it is because of this abnormal base pair here, what we will see, the loop formation takes place. And this C की अगर हम बात करें, it is the cytosine. What we will see, the cytosine is this. Now we have got third arm. This third arm is this. It is the variable arm. This is the third arm. It is the variable. It is also called as the vestigial arm. Now we have got this fourth arm. Fourth arm की अगर हम बात कर लेंगे, fourth arm, it is called as anticodon arm. It is called as anticodon arm, or it is called as anticodon loop, because this arm जो है, it consists of anticodon, and this anticodon जो है, it recognizes the codon on mRNA. For example, this is the mRNA, and this mRNA is in the direction 5' 3'. These are the codons. I will draw them with another this greenish, so that these codons become somewhat clear. These are the triplets. 3 words. These 3 words, they recognize what we will see. This, for example, this. And for example, if this is the A, if this is the AUG, now this anticodon, it decides the type of amino acid which will attach at the 3' end. This anticodon, it decides the type of amino acid which will attach to 3' end. Now the type of anticodon will decide the what type of amino acid will come and will attach at the 3' end of this transfer RNA, because anticodon has recognized the codon. Now this is the IUG. What will be the anticodons there? Adenine, it will be not this what will be called as this thymine. It will be the uracil. It will be the adenine, and instead of this guanine, it is the cytosine. So it has got this what we call, if this is the mRNA. Now this loop, it has got anticodon, and these anticodons, they recognize this codon, and which has got the genetic information for particular amino acids. And this is what we call as this region. अगर हम इसकी बात कर लेंगे, this it is called as anticodon arm, and it has got this again. It has got this what we call as this abnormal base pair, that is the inosine abnormal base pair, and it is because of this abnormal base pair no hydrogen bonding takes place. Now in case of this fifth arm, this is fifth arm, this is the fifth arm. See what we will see, this is also called as, what we will see, it is called as this fifth, it is also called as the D loop, because it consists of the what we call as this abnormal base pair, which is dihydrouridine. So what we will see, because it has got dihydrouridine or dihydrouracil, the function that it helps in, what does it is main function, what is the main function of this, that it helps us in amino acid recognition site. It helps in amino acid recognition site. Now it has got what we call as we see this, it recognize site, it helps us to recognize the amino acid recognize site, then this amino acid is attached what we called as the 3' end of this what we call as mRNA. This loop, it is also called as the T loop. This is also called as the D loop, and this is also called as the anticodon is there, because it contains, now this loop and this loop, they all have got these abnormal base pairs. What is this abnormal base pair? In this case, if we talk, it has got the pseudouracil, if we call, it has got the inosine, and if we talk of this, it has got the dihydrouridine, and it is because of these abnormal base pairs or modified base pair no hydrogen bonding takes place, and they give rise to these 3 loops. But what is the function of this arm? It activates amino acids, it activates amino acid attachment site. What is this? It is called as the anticodon. If this is the mRNA, it has got this code system like this, it is the code and in codon AUG, and we will see that it recognizes this what we call as this codon, which has genetic information for the particular amino acid. And this is the last one, this is called as the D loop, because it consists of the dihydroxyuridine, because main function of this is that it helps us in the amino acid recognition center. So what we have studied, we have studied that this clover leaf model, it is because of this what we call as the too much foldings, and it seems to be the double helix. It seems to be the, but actually what is this? It is the single stranded bond. It seems to be double stranded, but it is the single stranded structure, and it is the structure what we have discussed there, that is, it has got the 3 loops there. And what we will see, that main function of this is that it helps us in the transfer of, it is called the transfer RN, because it helps us in the transfer of amino acids. And these amino acids, they get attached at this what we call as the third end, that is called as the activated amino acid attachment site. So this was all about the structure of this what we call as the clover leaf model, which was given by the Philip Hawley in the year 1965 regarding the structure of this tRNA. In the next video we will try to discuss the last, that is the rRNA. Until then, goodbye, God bless you, thanks.